So I'm going to shift gear a little bit, not talking about architecture as you heard before, but about functional MRI. And so I kept the original title, Neurocoding of Computational Factors Affecting Decision Making, but I will give you actually a broader overview, talking about this, but also about other things. Just a general introduction. So the general goal of our team is to understand the normal and the pathological neural mechanism underlying decision making and reward processing. And to do this, we are actually studying three different topics. The first one concerns the functional organization of the prefrontal cortex. We did this early on in the field of cognitive control and task switching. We are continuing this work now on perceptual decision making and value-based decision making. And I will tell you some recent functional organization we found in the orbitofrontal cortex, in particular in this field. The second topic concerns the reward system. As you know, dopaminergic neurons innervate a number of cortical areas, such as the prefrontal cortex and the basal ganglia, and they are involved in a large number of motivational and cognitive functions. And so we are using fMRI and more recently intracranial recording to study the function of the reward system. The third axis of research concerns more the dysfunction of these two systems, the prefrontal cortex and the dopaminergic system, in a number of clinical populations, such as Parkinson's disease, schizophrenia, patients with prefrontal cortex lesion, and also more recently in pathological gamblers, in women with anorexia, and also in healthy aging, since we know that both the prefrontal cortex and the dopaminergic system are actually dysfunctional in healthy aging. So as I just mentioned, the main techniques we are using are fMRI and intracranial recording in humans, and also we did a few studies with MEG in healthy subjects. And finally, the fourth axis of research, which is parallel to the three axes of research that I just presented, concerns the genetic and gonadal steroid hormones influence on the reward system and the functional organization of the prefrontal cortex. So the general approach we are using, this is just a general slide that just makes links between different levels of observation. So what we are actually doing here is cerebral activation in given task. And the idea is to make links between this system level of organization with electrophysiology, as recorded in monkeys in particular, and the link with behavior through the use of neurocomputational models. So this is an important approach, which is called now model-based fMRI. And the idea is not only to look at brain activation in a given contrast when you compare condition A versus a control condition B, for example, but to have a neurocomputational model of the mechanism that make a link between the behavior and the function of the region. So the idea is that a specific region is supposed to do a certain computation, and it's not just a mapping of areas that are activated in a given contrast. It's more, it has to be viewed more as a computation that is done in the network that is identified. So the general question is to know how do we make a choice in general, and this is a very large and question. And so we are doing this in very restricted type of decision-making experiment, where one critical stage is called the valuation stage. The idea is that we attribute a value to different actions or different options, and that we maximize the option. That is, we choose the option that has the maximal value. After we make a choice, and then we have an evaluation of the outcome that is potentially used for learning. And this is a very general presentation, but I will give you a few examples of some results that we had in this domain of valuation. So how does the brain compute values? This is a very large question. And, here is just a general slide that presents different type of modulators or computational factors that are involved in computing these value signal. This is coming from a number of economics theory, such as prospect theory, that is displayed here. But we also know that, for example, the delay before the reward obtention is involved in this value signal. and there is also a number of social modulation that influence value. So today I will actually talk about three fundamental goals. The first one concerns the original title of the talk, which is how the brain encodes reward-related parameters or computational factors. And I will specifically talk about reward probability encoding and uncertainty in the brain. So the idea here is that reward value is dependent upon a number of factors, such as the probability of the reward, the amount, and the delay. And this has been described in a number of computational models. And we will use this model to see how specific regions are modulated by this value function, in particular by the probability of the reward or the delay of the before the reward obtention. The second goal concerns the identification of common and specific regions that are involved with different rewards. So the idea here is that sensory inputs and physiological needs at some point to be integrated, and this is the idea that there might be a common reward currency network in the brain that allows us to decide between different options of different nature. So the idea here is that there might be a common network that computes this common reward currency. But there is also another idea that might be that more specialized area might be devoted for specific types of rewards. So this is driving a number of studies that I will present rapidly today. And so the idea here is a bit to revisit or to fractionate the reward system, which is seen as a whole usually, but that there could be some components that are actually more specialized than what is originally thought. The other idea concerns also the punishment or the aversive domain, because there have been a number of studies from, for example, the Ikosaka lab that reported that dopaminergic neurons are not only responding to classical prediction reward error, but are also involved, as I will show you, with what is called a salient prediction error that also respond to punishment. So this is actually contradicting the classical view that midbrain dopaminergic neurons are more involved with reward alone. And so we will study at the system level how we can use previous models on prediction error to investigate where in the brain we have a computation of salient prediction error. The third goal concerns more the relationship with economics and behavioral economics. And here is just the general idea that it's important also to study value in the context of social decision making. So there have been a number of studies, for example, using hyperscanning, where people are connected and are scanning two different scanners, for example. What I will show you is some recent studies that just concern this area, and this is one topic where we are actually moving to that concern more group decision making and social decision making. So again, this is a general slide that shows you the approach that we are taking, that is model-based fMRI, that just remind you what I was telling you about. That is, we are looking at where in the brain a specific computation is, which regions are engaged by specific computation. And this is just represented here by computational models that give the value of, for example, a given option if you have a choice, or just in a given situation when you have no choice, for example. So the first study I want to tell you about is actually based on a monkey electrophysical study where monkeys were conditioned, and there was different type of stimuli like this that were presented with different reward probabilities. And in this study, what was shown when the Schulz group actually found was that there was two types of response from midbrain dopaminergic neurons, one which is transient and one which is sustained. And what is interesting is that, just summarizing these results, which are already classical, is that there might be three types of theoretical measure encoded by these neurons. The first one concerns the expected value, or the utility, that increase with the reward probability at the time of the condition stimulus. The second one concerns the prediction error, that is encoding the difference between what is expected and what is actually delivered, and that is decreasing with reward probability at the outcome. And the third theoretical measure concerns uncertainty, which is maximal for probability of 0.5 and is less pronounced for probability of 0 or 1, for example. So based on this study, we designed a first fMRI study where we look where in the brain we have an encoding of this transient and sustained mode of activity. And we search with fMRI where, so the advantage is compared to electrophysiological recording, we can identify not only midbrain region, but also the whole brain. So if there are specific postsynaptic dopaminergic projection sites that respond more to the transient and the sustained aspect. In this first study, we use only monetary reward because it's a reward that can be easily modulated in, concerning the intensity and the probability. So just to make it, so the idea was to have a task that is very close to the one used in monkeys. And in the monkey paradigm, they actually recorded from dopaminergic neurons after over-learning. So here we actually gave the actual probability of being rewarded explicitly, unlike in a casino. So the subject knew that, for example, this slot machine has a 25% chance, as indicated by the red portion of the pie chart, to be rewarded by $20, and 75% to be not rewarded. So we had a number of different slot machines like this. I'm not going to go through all the detail of the paradigm, but the idea was to vary orthogonally both the probability and the magnitude of the reward. And then there was the spinners that roll around during a long delay period of 14 or 15 seconds, and then the outcome, which was either no reward delivery or the reward delivery. So in this first study, the main results were that we were able to replicate monkey midbrain dopaminergic studies that I just told you about, which is that there was an increased midbrain activity at the Q with reward probability, a decrease midbrain activity at the time of the outcome with reward probability that is supposed to encode prediction error, and also a signal that was recording during the delay period with reward uncertainty. What is interesting is that concerning the postsynaptic sites that are shown below is that there was a distinction between sustained activities coding the uncertainty signal in the ventral striatum, putamen, and extending in the claustrum, and more cortical region, prefrontal region, encoding the predicted value as well as the prediction error. So this was the first study that tried to decompose the different theoretical measures I was just showing you. So the idea will be that the lateral prefrontal cortex may process the expected value as well as the reward prediction error in this study, and that the ventral striatum may reflect more the expectation of reward information in terms of information theory, since we know that this curve might reflect entropy or variance. So obviously there are problems with, so if I come back to this timing, is that the delay between the Q and the outcome is very long, and this was done purposely because with fMRI, the idea here was to distinguish the transient aspect at the time of the Q and the outcome from the sustained activity. That's why we had a very long delay. But it's also very different from the monkey study, which used only 2 seconds between the Q and the outcome. So we thought to use intracranial recordings in a new, so the idea is to use intracranial recording to investigate other regions that maybe we didn't identify with fMRI. This is just to show you a previous study that actually shows that the orbitofrontal cortex is known to be involved in decision-making under risk. And this is why we ask whether the orbitofrontal cortex is also encoding uncertainty in the human brain. So we adapted the paradigm that I just showed you by giving, again, slot machines to the subject. Unlike in the previous paradigm, where they were giving explicitly the reward probability, here they were shown fractal on top of each slot machine, and they had to learn whether the outcome of each slot machine was whether good or bad. That was more a learning paradigm. And so in this study done by Giovanna Vanni Mercier, we looked first in the orbitofrontal cortex whether there was an encoding of reward uncertainty. This is just summarized here. Maybe it's not very clear, but this is recording from 6 patients with orbitofrontal cortex implantation. And this is data analyzed by Yong-Sung Lee, who is doing his PhD. And so what can be seen here in blue for probability of 0.5 is that at the time of the outcome here, and this is also displayed in this 2 time interval, you have actually an encoding of reward uncertainty that is true not only for rewarded outcome, but also for unrewarded outcome. So the idea is that uncertainty might be encoded in the orbitofrontal cortex at a more precise temporal level than what we could observe with fMRI. In the same paradigm, we actually recorded also from patients with hippocampal implantation. And again, we found that at the time of the outcome, so the outcome here is in fact the time when the third spinner stops, when the subject actually know that they are going to be rewarded. There is again an encoding of the reward uncertainty. So we are doing actually now more refined analysis to know whether we can actually direct, for example, a timing property from these 2 signals to know whether the orbitofrontal cortex is first before the hippocampus. So just an interim summary from this study that look at computational factors such as reward uncertainty and probability. So concerning the mean brain, we found that we can indeed reproduce the results that were actually shown in monkeys. And more interestingly, we can distinguish different projection sites that respond differentially to expected value and prediction error, as well as uncertainty. Concerning the local field potential studies that I just showed you, we might thought about this uncertainty signal as being fundamental for underlying, in particular, the role of the hippocampus in attention-based learning and probabilistic classification, because it might signal when the uncertainty is maximal that learning new learning need to be done or encoded. And so, from a more general idea here is that the LFP recording might be a complementary and valuable approach to monkey recording and to human fMRI. So I'm shifting now to the second goal that I was presenting that concerned not only different type of reward, but also punishment. And in this study, which was recently published in Cerebral Cortex by Elise Metero, we looked at, it was based also on an electrophysical study published in Nature in 2009 by the group of Ikosaka, as I was mentioning before, that showed that there might be a salient prediction error in addition to classical reward prediction errors that have been reported in this neuron. That is, these neurons are excited not only by unpredictable rewards, but also by unpredictable aversive events. So this is an important finding because this actually extends the role of midbrain dopaminergic neurons to punishment. And so the question we addressed here was to know whether specific human brain structures which receive from dopaminergic neurons actually code a salient prediction error, and whether this type of signal depends upon the reinforcer type. So the reinforcer here were juice, which was either positive or aversive, which was really delivered in the subject's mouth during scanning, money, and aversive images. So the exact paradigm is actually very close to the monkey study, and the idea here was there was always a maximal uncertainty, that is probability of 0.5. So you have cues that predict these 4 types of reinforcer, juice, like apple juice, salty water, which is aversive, money, or aversive images. And this is just to show you the pond that we used to deliver the juice. This is one example of a given trial where you have a cue that is associated to the outcome. And we use computational modeling to model both the prediction error and the predicted value. So I'm not going to go through this. These are classical models that are used in the field. The idea here is that we will look for responses that are consistent with the salient prediction error, that is, that respond positively to reinforcer not only for reward, but also for punishment, while the reward prediction error will predict that you have a negative prediction error for punishment. And so we analyze the data by looking both at the anticipatory period, which concern this period during the delay, that is modulated by the predicted value, V, and we look also at the prediction error at the time of the outcome, encoding the salient prediction error in a number of combinations that we can do between the different type of referencer. So just to be clear, the first result here concerns the anticipatory period. So both for apple juice and money and for salty water or aversive picture, we found that the ventromedial part of the prefrontal cortex is encoding a salient prediction error, which is a new finding, actually, during this anticipatory period. And at the time of the outcome, concerning the salient prediction error, we found different contribution of different regions according to the type of reinforcer. So what we call gustative salient prediction error here concern only the juice, whether it is aversive or positive. And we found that the insula, the anterior cingulate cortex, and the putamen responded to this gustative salient prediction error. We also found that there was an encoding of what we call salient prediction error that is primary because it was responding only for aversive features, apple juice and salty water, but not for money. So just to summarize this study, we showed that during the anticipatory period, the ventromedial prefrontal cortex encodes the predicted value regardless of the type of reward or punishment, and that we can distinguish between different brain networks that engage the striatum, anterior insula, and anterior cingulate for the gustative salient prediction error, while the amygdala is more responsive for primary salient prediction error. So this is just an example that shows that it's possible using this model-based fMRI approach to characterize how we learn stimuli reward or stimuli punishment contingency by demonstrating that the network encoding the salient prediction error depends upon the reinforcer type. Again, concerning this same goal of distinguishing between common and specific neural substrate for different types of reward, we did one study where we actually had no learning. This was, again, based on this idea that at some point there is a need to integrate sensory input and physiological need for encoding a common reward currency. And so we look for potential dissociation within the orbitofrontal cortex according to reward type, because there was actually a review that suggested that there could be an encoding of more abstract reward in the anterior part of the orbitofrontal cortex and more posterior part of the orbitofrontal cortex encoding more primary rewards. Which was just based on the meta-analysis with no direct evidence for this. So we actually tested this hypothesis using very simple paradigms that actually is very close to the slot machine paradigm that I told you about, where we varied both the probability, the type of the reward, and also the magnitude or the intensity of the reward. Unlike in the slot machine experiment, there was actually an additional discrimination task that the subject needed to do during the scanner just to maintain them in the task. And we looked at the result at the outcome period. And we also use, I'm not going to detail too much about this, but rating from the subject done on every trial to look for brain correlates of this rating that is encoding actually that is a measure of the value of the stimuli. So this is a design that shows you that there is all the range of probability, the two types of intensity for the two types of reward. And the first results that are coming from this study are the regions that are common to the two types of reward, that are reflecting a classical reward network that engage the striatum, the midbrain, and the cingulate, as well as the insula. And so these are what we call the classical reward network. More interestingly, we found also a nice dissociation between the anterior part and the posterior part of the orbitofrontal cortex. The most anterior part encoding more secondary reward, while the posterior part and the medial part encoding primary rewards. We also found a response that was robust in the amygdala that was more responding to primary reward as compared to secondary rewards. So taken together, this study shows that there might be dissociation between the anterior and posterior part of the orbitofrontal cortex that may be interesting to relate to the ontogenetic and phylogenetical development of these brain regions, since we know that the orbitofrontal cortex, the most anterior part is more recent phylogenetically than the posterior part. We also showed that there is like a common neural currency that is common to the two types of rewards that is reflecting more in the classical reward network. And this finding in the orbitofrontal cortex might be also related to more classical theories and experiments that, for example, looked at the functional architecture of the lateral part of the prefrontal cortex that also propose a gradient of complexity from the posterior to the anterior part of the lateral prefrontal cortex. So we extend this in the domain of the value domain in the orbitofrontal cortex. So one question we tried to address more recently was to know whether this result could be extended to other types of reward. So here there was just a paradigm. We were always limited with the timing of one single fMRI experiment. So we only compared two types of reward. But in a meta-analysis, Guillaume Sescus, we actually performed the previous experiment, also did a meta-analysis with a software called ALE on 87 studies that included a very large number of subjects for not only reward and erotic stimuli, but also with food stimuli. So this is the result that should be published in the Neuroscience and Biobehavior Reviews in 2013. So I'm not going to go through all the activation pattern here, but the main idea is that we confirm that the striatum and the ventromedial part of the prefrontal cortex are engaged by the three types of reward: money here in green, food in blue, and erotic reward in red. And we reproduce some of the results from Guillaume done in his previous fMRI study. That is, money, when compared to either erotic stimuli or food, are actually engaging more the anterior part of the orbitofrontal cortex. But we didn't find the same dissociation. That is, we did not reproduce that erotic stimuli were engaging more the posterior part of the orbitofrontal cortex. They were engaging more the amygdala, as we also shown before. And interestingly, they also engage regions that are involved with body representation in the extra body area. We also found some confirmation of what we actually already know, that the insula is more engaged by food reward as compared to erotic or money, for example. So this is not only a mere confirmation of the single fMRI studies that I showed you, because there are always limitations in this type of meta-analysis, but this is complementary to what I showed you before. How am I going to, with time? How much? 5 minutes only? Okay. Okay, yeah, I don't know what I'm going to present. So far I told you about value representation only in quite passive experiment. In another study, we actually studied value-based decision-making, which is more related to the neuroeconomics domain. So in this first study, the idea, again, as I was mentioning before, is that a decision is made after evaluation stage. And classical economics theory actually do not make a distinction between the type of costs. So the idea is that when you make a cost-benefit evaluation, the choice is made regardless of the type of cost after this valuation stage. So in this study, what we looked at is actually whether there is a single valuation system in the brain, or whether it's actually depending upon the type of cost. And the cost here would be referring to delay or effort. So this study was based on a number of studies, not only fMRI in humans, but also animal study, concerning what is called the delay discounting paradigm. So briefly here, for example, you have subjects that are presented with a choice of, for example, $20 now versus $40 in 30 days, and this delay and amount are varied. And what has been found by Cable and Glimcher in particular is that there is a valuation system that engages the VM, ventromedial prefrontal cortex, the striatum, as well as the posterior cingulate. And what they actually imply is that there is a single valuation system in the brain. However, this was not tested because they only use one type of cost, as I was mentioning, which is a delay. And it's still unclear whether the same valuation system will be engaged also with what we could call effort discounting. So this is the goal of this experiment, where we actually distinguish delay versus effort discounting in the brain. So we use primary rewards and not money in this experiment because with money there is actually a problem, because the subject do not experience a reward at the time of the delivery, obviously in the scanner, and they do not really experience the delay either. So we did a cost-benefit decision-making paradigm with these two types of costs that I was mentioning. The cost being the delay in the delay discounting task with different level, and the benefit being the fact that the fuzzy image presented here was delivered for shorter or longer time period. So the subject had to accept or reject the choice, and we look at the evaluation period during the time of the decision. We did a very similar paradigm for the effort discounting paradigm, where this time the subject had to integrate the potential benefit of seeing the image in clear and the cost of pressing with the hand grip and making an effort that is presented here on like a temperature type of level. So maybe I'm not going to go through all the behavioral results, but we could actually model the subjective value associated to these two types of costs using classical economics type of model. And we showed first that concerning the delay discounting, we could reproduce what has been found before with money. That is that the ventromedial part and the striatum are engaged with the subjective value of the delayed reward. And so this is concerning the delay discounting option. But crucially, concerning the effort, there was actually it was a different network that was engaged with effort discounting. And it was actually a negative correlation with the subjective value of the effortful reward that engaged the insula and the anterior cingulate. So the subjective value is, as I said, just a negative correlation in this network that is reflected by the fact that these regions are engaged by a higher level of effort, as you can see here. So just to conclude on this study, we found not one single valuation system in the human brain, but that two different networks reflect in opposite fashion delayed reward and future energetic expenses. So we are extending this to psychiatric population. I don't know if I will have time to mention this. So just to conclude on this study, to try to integrate all what I've shown you, this is from a recent review from Matthew Rushforth. And what he proposed is that based on the lateral orbitofrontal cortex that represent the value, there will be two distinct roles that could be possible for the ventromedial prefrontal cortex. It could encode either the value expectation based on the lateral orbitofrontal cortex information, or a value comparison. And then this message, so for example, the value expectation could be done, for example, in passive experiments, such as the one I showed you at the beginning, and the value comparison would be done more in value-based decision making. And if you, this is just a slide summarizing all the results I showed you with fMRI that are consistent actually with these two hypotheses. So we, just to remind you, what we found in the Meteor paper is that the medial part of the orbitofrontal cortex encodes actually the value expectation during Pavlovian conditioning, both for reward and punishment. We also found that in this meta-analysis, the ventromedial prefrontal cortex is encoding the experience value at the time of the outcome, not during the anticipation, and that there are also a dissociation between the anterior part according to the type of secondary or primary reward. We also found that uncertainty is encoded in the orbitofrontal cortex, and what I just showed you is that this same ventromedial prefrontal cortex is engaged at the time of choice during delay discounting, but also in other paradigms that I don't have time to show you here. So I don't know if I have time for the third axis of research, which concerns more the social decision making. Maybe I show one example, and then I will conclude. So this is actually from the study we did with Marie-Claire, and Elise Meteo was also the PhD student who did this study, where we looked at the effect of audience on two types of prosocial behavior. So the idea here is that we are studying now a different type of choice and very abstract decision that are more in the domain of moral decision. And what is known from the literature is that subjects are actually, they show altruistic inclination, relying on moral values and preference, that they are also averse to inequality and so on. And there are recent economic theories that prosocial behavior that combines heterogeneity in individual altruism and greed with concerns for social reputation or self-image. So one recent hypothesis that has been formulated is that actually humans exhibit social preferences for prosocial behavior, not because they are intrinsically good, but because of concern for self-image or audience. So this is, we can distinguish, in fact, prosocial decision or altruistic donation, for example, can be attributed to different motives. The first one is an intrinsic motivation, which would be like the intrinsic motivation of being good, so that the private preference for other well-being, which we call moral reward. There is also the motive which is intrinsic, such as any material reward or benefit that are associated with giving, such as, for example, getting money in some country when you give blood. You can be paid for this. And the last one is the one we are studying actually here, is concerning the audience or the image motivation, which refers to the individual's tendency to be motivated partly by how others perceive us, so which is more a social reward. So the idea is that prosocial behavior depends not only on the ability to be motivated by how others perceive our behavior, image motivation, but also by the social or moral value attributed to the recipient of monetary transfer. And so we ask how image motivation modulates choice behavior, as well as the neural evidence for the existence of concern for these two types of decision. So we had a paradigm where we had subjects had to make choice that were either public or private, which are represented here, private by a lock and public by some eyes. They had, again, a cost-benefit decision that had to be done either on a positive organization called Resto du Cœur here, or negative organization, which is a Groupe d'Action Royaliste. And there are different levels of money that were varied, where if they accepted the choice, they could get a certain amount of money, and I mean, the organization could get a certain amount of money, and they will lose a certain amount of money. In the negative organization condition, it was similar, except that the organization could get some money, but the subject should actually earn some money, not lose some money. So this is just the time course of trials, and the behavioral results are displayed here, actually are quite nice. As you can see, this is the percentage of accepted trials, and the subject loss are represented here. As you can see, they are going to refuse or accept less tries when the loss are higher, and they are also accepting more to give when the amount going to the positive organization is increasing. There is a like a very symmetric type of behavior that was obtained for the negative organization. That is, when the subject gain is higher, they actually accept more, this is shown here, for example. And when the amount going to the negative organization increase, they actually accept less to give to the negative organization. What is interesting is the actual audience effect that we observe, which is also shown here. So in green, in pink, you have the positive organization, in blue, the negative one. And private and public are shown here. So this is what we call the audience effect. That is, in public, subjects are going to give more just because they know that they are observed. And the private situation is just the converse that is observed for the negative organization. That is how money can buy social moral values. So subjects, in a way here, can be described as selling their own soul, that is a moral value, to earn money when they make that choice in private instead of public. So it's quite a nice behavioral finding. So just to go quickly over the fMRI results, what we found when comparing just a public versus a private decisions, that is reflecting the audience effect, we found a network that is engaged with mentalizing, that is known to engage the cingulate as well as the superior temporal gyrus, which makes sense because this network is known to be engaged when people attribute attention to others and are known to. So we are showing here that it's engaged when they are potentially paying attention to others. I don't know if I go through all these results, but just to conclude on this study, for the lateral prefrontal cortex, we also found that the subjective value of the selfish and the amoral decision are encoding in actually the same region. The slide before was showing actually common, distinct region for the two types of positive and negative organization. So what is shown here is that when subjects reject the choice in the positive organization in private, so they make selfish choice, or when they or when they accept in private for the negative organization, they engage the same lateral orbitofrontal cortex. So this reflects that this lateral orbitofrontal cortex is encoding subjective value both for selfish decision for positive organization and amoral decision for the negative association. So to conclude on this this social reward, we found that the mentalizing network is engaged more when subjects make public choice than in private. The prosocial behavior engages also the ventromedial prefrontal cortex. I didn't have time to show you this result. The lateral orbitofrontal cortex engage more, are more engaged by rejecting to transfer money to charity and accepting to earn money at the cost for own moral value. So this is direct evidence for the validity of prosocial theories that combines the heterogeneity, individual altruism with concern for social or image motivation. And it's the first identification of the brain circuitry sensitive to concern for self-image in the context of prosocial or amoral behavior. So I'm going to go through all this. This is another study looking at group decision making. I don't have time to go through this. Maybe to conclude, so there was the last three axes of research I was mentioning at the beginning concerned the application of this type of paradigms to clinical population. So it's just maybe to conclude on this, just to give you an overview of some recent findings that concern clinical population. Or here is healthy aging, where we combine actually multimodal imaging, fMRI and PET, to look at the same reward paradigm that I was presenting you, that relate age-related qualitative change in the interaction between midbrain dopaminergic activity and the reward system. In another study, in a number of studies actually, we also investigated how the specific dysfunction of one motivational system is dysfunctional in different groups of clinical population. For example, here, this is one study done by Romain Lignol, who is doing his PhD, and Guillaume Sescos, and where we look at risk aversion. And what he found is that pathological gamblers, so this is a classical type of prospect theory curve that shows that subjects are over evaluating low probability when they make choice like this between a sure option and risky option, and underestimating high probability. So one actual hypothesis formulated by Kahneman and Swirsky, who won the Nobel Prize, was that their prediction would be that pathological gamblers might actually overestimate even more than control just low probability. And in fact, it's not exactly what we found, because we found a shift of these functions that concern not only low probability, but also high probability. So it's just to give you an example of a study we are doing in pathological gamblers, where we also apply the paradigm I showed you, comparing monetary reward with theoretic reward, where we had one quite interesting finding in the posterior part of the orbitofrontal cortex. Since we reproduce the finding from Guillaume that aortic stimuli engage more the posterior part of the orbitofrontal cortex, but that gamblers actually treat money as a primary reward, since there was no difference between these two types of rewards, and they were engaging more the posterior orbitofrontal cortex, both for monetary reward and for aortic stimuli. So we are using the same type of strategy using the paradigm I showed you, for example, before, in patients with Parkinson's disease, now with impulse control disorder on/off L-DOPA, and this is ongoing work. We also are doing a number of studies in patients with schizophrenia, one just recently published in Biological Psychiatry concerning the comparison between patients with schizophrenia and healthy aging. We apply also the same strategy to the one I showed you in pathological gamblers in these patients. And we are also studying women with anorexia in a type of loss aversion paradigm that are comparing between, for example, monetary rewards with juice reward. So I don't have time to go over all this, but we are doing this also with fMRI. Okay, so just to summarize, and maybe to sum up, so the last axis of research, I'm not going to go through all this, but the idea is to look at the effect of functional polymorphisms in genes that affect dopamine transmission, and also how gonadal steroid hormones, such as estradiol in women and testosterone in men, affect value-based decision-making as well as reward processing. So we are doing a number of studies like this, where one original one was published in PNAS recently, and another one in 2009. And so this is just to conclude the take-home messages. The first concerning the functional organization of the orbitofrontal cortex that I showed you, that concern primary versus secondary reward and the computation of subjective value. The second axis of research that is actually the topic of the talk, but that I extended it to a number of computation of factors that are involved in reward processing, such as reward probability, uncertainty, delay, and effort. And we are characterizing also different components that are involved in social interaction. Finally, in the domain of clinical population, we are identifying the dysfunction of the reward system and decision-making system in a number of neurological and psychiatric disorders with this model-based fMRI approach that I was showing you, that allows us to identify new and different phenotype characteristic of psychiatric disease. And the final axis of research that I didn't have time to detail, that concern more the genetic and hormonal influence that are actually an important challenge to identify in the future. This is the team in its current form with the current postdoc and PhD students, and I want to thank particularly the previous lab members for doing the studies that I presented today. Giovanna Vanni-Mercier, who left the lab, as well as Guillaume Sescoux, Philippe Domenech, Charlotte Prévost, Elise Météro, Julie Thomas, Jérôme Redouté, and Xavier Caldufero. And also for the task I presented here, Romain Lignol, as well as Romuald Girard. And Pierre Vidot for some of the study done with Romuald Girard that I didn't have time to present. And finally, it's just to mention a book I edited with Léon Tremblay, and just to advertise it, that is a big summary of different fields of research that concern reward and decision making, published by Academic Press in 2009. Thank you.